I'm going to move on to the third part of this module. So here we are, genetic technologies. Does artificial manipulation of DNA have the potential to change populations forever? So here we go. We're going to discuss the uses and advantages of current genetic technologies. We're going to talk about reproductive technologies, cloning, recombinant DNA technologies, benefits to agricultural and medical and industrial applications, effect on biodiversity of agricultural biotech, influence of social, economic, and cultural contexts, and our inquiry question is, does artificial manipulation of DNA have the potential to change populations forever? Okay, recombinant DNA technologies. Let's have a look at how we, this is essentially recombinant DNA technologies. It's a method to join together DNA from two different species in order to produce new genetic combinations. So transgenesis is the process through which we introduce exogenous genetic material, which is like material from outside the person, into another organism. And it is performed so that the organism can exhibit a new trait. And remember, DNA is ubiquitous, it's the same across organisms, so we can insert new genes into genomes. And the way we can do it is we can either do it through plasmids, retroviral vectors, or DNA microinjections, which is like a fine glass needle. So this can actually be done for agricultural and medical applications. So what do? We begin by identifying a gene of interest. We extract that gene right there. We take it and we're going to deliver it, and we're going to put it in a culture, and then we are going to lead to, and then we're going to grow it so that we can have a genetically modified crop right there. So how do we make a transgenic organism? The first thing we do is we identify our genes, then we get large amounts of a gene of interest, and then we insert the gene into a target organism. The first technique to get to identify our genes is gene sequencing. This is what we looked at DNA sequencing in module 5, so I'll just skip through this, but it allows us to gather single nucleotide information. We can identify genes with useful traits, and we can use that. The second step is basically using PCR. Either PCR or gel, either PCR and then gel electrophoresis. So PCR to amplify the DNA, to get lots of copy of the DNA, and then we're going to use gel electrophoresis to visualize the DNA to check that we have the right type. So we're going to get large amounts of it, but we're going to check that we have the right type, and then we're going to insert it into a target organism. We can do it through restriction enzyme cloning, so what we looked through in that earlier example, where we basically get the target gene, we get the plasmid, and we basically put it together and we get a recombinant plasmid, and we're going to put it into a bacteria. We can also use transfection, which is basically we're using a virus as a vector. We're going to use a virus as a vector to put the gene into the target cell. Remember, viruses, they don't have their own machinery to produce proteins. They will go into what is going to happen is we're going to use the virus as a vector. So we're going to put a gene into the virus. We're going to send the virus into the target cell, and the virus is going to hijack the machinery of the target cell. It's going to then lead to the target cell. It's going to ask the target cell, basically force it to then make the proteins of that specific gene that we wanted to make, because that's what viruses do. But this time, instead of having the viral proteins in there, we're going to have the genes that we want. And finally, microinjection. We use a very fine needle, and we just, because the cells are small, and we just inject the DNA. Okay, here we go. That's the whole process. I will leave that for you to have a look at. Let's talk about some examples. So agricultural example, BT cotton. BT cotton, so basically the Bacillus thuringiensis produces Bt toxins, which are insecticidal, so they kill moths, beetles. And what we have done is the Bt toxic gene is inserted into cotton crops, because when pests eat the crop, the toxin is released and the pest is killed, and that reduces the need for farmers to actually use insecticides. The medical example of E. coli that we just talked about when we talked about recombinant DNA. So we know that insulin is an essential hormone which aids in the regulation of glucose metabolism, and inadequate production of glucose can lead to the development of diabetes. And what we do is we insert human insulin into E. coli, which is a bacteria, and we allow it to produce large amounts of it. We then take it, we purify that protein from the bacterial cells, and we use it to treat diabetes. Here we are. Recombinant DNA technology. So gene therapy is the correction of disorders. So we can actually correct disorders by introducing changes into the functional genes, and we can introduce healthy genetic material. The cell and its offspring can then have restored function, and we can insert using viruses, gene guns, or inorganic nanoparticles, and it can happen either in vivo in the living thing or ex vivo. CRISPR-Cas9 is a technology that actually edits the system for accurately introducing point mutations. So we introduce point mutations to fix something in the genome. So it has two parts. It has the guide RNA, which contains the nucleotide sequence, and it has the Cas9. The guide RNA is basically going to contain the complementary sequence that we want to edit, and the Cas9 is an endonuclease. It's actually going to cut the DNA so that the guide RNA can get in there and fix it up. ELISA is enzyme-linked immunosorbent assay. So what that is that it's a biochemistry tool which detects the presence of antigens in a liquid sample. So what it does is it's a diagnostic tool which exploits natural antigens, antibody interactions. So within our bodies, we have this antigen-antibody interaction, and what happens is that the levels of biomolecules in a sample can be measured by linking levels of antigens or antibodies to a fluorescent readout, and it's useful for forensic epidemiology, and we can trace the spread of infectious diseases using this. So all our cells have antigens, and those are like our body antigens, like our self antigens, but every time we encounter a virus or a bacteria, they have their own antigen, and the body recognizes that as like an outside thing, right? Like it's a non-self particle, and then the body has its own antibodies that it uses to latch onto the antigens and identify those antigens as non-self. But these are the three right there. I would recommend watching like actual videos of how these technologies work. You can find some really good ones, because that will give you like a visual of how they work. I know we're going a bit over time here, so I'm going to finish up. I'll leave just practice questions for you. Last bit. Here we are. So agricultural techniques and benefits. So techniques include selective breeding, artificial pollination, transgenesis. Benefits are that it leads to the creation of crops and livestock with favorable traits, high yields, high nutritional value, better temperament, and also reduced environmental impact. They can create organisms which do not require insecticides or herbicides, and it increases food security. In terms of medicine, there can be cloning, gene sequencing, gene therapy, CRISPR that we've looked through, and ELISA. Benefits are we can have personalized medicines. There can be preemptive diagnosis, treatment of genetic diseases, potential cures using like CRISPR-Cas9, and gene sequencing, improved diagnostic tools, cheaper, faster treatments, and creation of molecules which can be used for medical treatments like insulation for diabetes. Industry techniques, gene cloning, sequencing, transgenesis. So benefits are we can have increased speed of chemical reactions, more efficient industrial processes, creation of many everyday items, and creation of organisms which produce industrially significant products like biofuels, biomaterials, and energy. I'll leave the positives and negatives for you. They're very similar to what we have discussed. I will also leave the social, economic, and cultural context for you, because this is just putting together what is the influence of social, economic, and cultural context on biology, on these biotechnologies. So that includes, like looking at the big picture of countries, law, environment, religions, economies, and bringing that down to the science and the students and the relationship between the two. So that's there.